Okay. I'm going to back down a bit, and we'll get you enough slack so that you can turn around and see Herc.
Sounds good.
It might involve more coming down
Okay.
on your part because I'm backing down slope. And you're going to start to drift back towards the ship.
Okay, I've got 30 meters. Do you want me to keep coming?
That's fine. Just watch your delta from now on.
Okay.
I'll stay near the bottom and... And you can do a Doppler reset and switch to Doppler base nav if you want.
Yep, thanks.
I'm going to stay close to the seafloor from now on. And you can turn your auto head on.
Okay. Auto head is on. What just happened?
Yeah. Just the switch to solution, to dead recon solution. Can you switch back to USBL or just do your, just get me a better position? I'm gonna hold still for a second here.
Yeah, same.
Yeah. Was that just a bad,
Well, do you have Doppler lock right now?
No. Oh, yeah, I do. I have 4 beams.
Okay.
So maybe it just needed a reset.
So we try again?
Yeah. And then just do a reset. Okay, I don't know. Can you just go back to USBL? Once we get the vehicles configured properly, then once we get it so we can see the vehicles and everything's stable, we can troubleshoot it.
Okay.
I have a view of you if you want me to bring my heading around a little bit.
Sure, just give me 1 second.
Yep.
I want to see where everything is first. Okay, that's lovely. Yeah, okay, do your thing.
Okay.
I'm going to come up just a little bit
Okay.
so that I can see anything at all.
So much stuff swimming. Are they swimming?
Okay, I
or
might
is
need
it
to back
just
down
sediment?
more for you to come around.
Yeah, okay.
Yeah.
Yeah, you just have to periphery my... All right, I'm getting you in view here.
Okay.
There we go.
Okay.
Guys, once we move away from this, can we trigger a Niskin?
Can you stand by, Piero? We've got a bit of, we've got some operational stuff to take care of here.
Yeah. Whenever you're ready. No
Video, what's going on with Atalanta HD there? Okay.
That's a great question, sorry.
Can you bring up the focal and look at the fiber levels there?
Yeah.
Yeah, I adjusted iris and that started happening.
Yeah.
They look fine?
They're fine.
Okay, great. Cool.
Yeah.
Okay. I just wanted to make sure it wasn't something to do with the vehicle health. Okay. I can't do that right now because I don't have auto XY. Okay. Shall we figure out what's going on with...
Yeah, I think we should just try again.
Yeah, cool.
Okay.
Sounds good.
So, why is it jumping?
So we definitely need the Doppler to be working right to do this survey.
Let's see, how high are you right now? You're 5 meters.
I'll back down. Sorry, I'm stretching you out a bit.
That's all right.
Just makes it hard to manage the tether.
I just don't know why it's jumping all the way over there.
You might have to come up as I get closer to you.
Yeah, I am
Okay.
coming up a bit.
Okay, you can manage the tether however seems reasonable.
Okay.
And I should be good to stay here.
Okay.
I don't think there might be a little more shift to Atalanta, but not too much.
Yeah, okay.
Do you mind if I test again? I want to see if it jumps to that weird pixel over there.
Go for it. Oh, it's just like a pixel that it's going to?
Yeah. Yeah, it's jumping to that for some reason.
I
I'm
could probably
gonna come down a bit.
Okay.
To like reset it.
Huh, I see the wrap in the tether.
Yeah. How did that? I thought we got the wrap out though.
We didn't take anything out.
No.
We left it exactly the same as it was.
Okay.
All we did was get a loop out.
Okay.
I think it needs a, the vehicle to turn to counterclockwise once. I'm gonna wait until we're done.
Yeah.
One turn to counterclockwise is just gonna make it sit at a -5, so it's not -0.5, so it's not really a full. You just sort of orbit this like half wrap.
Yeah.
You want to be on the correct side of it.
Can you turn it off and then turn it back on, the Doppler?
Yeah, I can.
I'm wondering if that would like kind of get rid of that and then put it, give it new data for where it actually is.
I don't think that turning the Doppler off will change that. I think that restarting the software is more likely to change that.
Okay.
How do you, I don't know whether you guys typically do that or not do that.
I think it's fine.
Okay. Yeah, I don't think there's any like sub maps you're gonna have to reload, so that's what usually takes forever.
Okay.
I'm happy with that. pixel is still there. We may need to do the full, restart the full nav. I can try power cycling the Doppler.
Well, let me try giving it a new source first to see if it jumps again.
Okay.
Yep, still doing it.
Yeah, okay.
So yeah, I'll just shut it all down and
Yeah.
reboot it.
Sounds good.
I'm just doing a bottom gauges.
That's awesome. Thank you.
Main is sitting at 2.
Beauty.
It's in a different place.
Yeah, let's see.
Oh boy. What is that all about? All right.
I'm ready for you to give it a go if you want.
Jump to the new location.
Okay. Weird.
Yeah.
Okay, I'll reboot the Doppler.
Oh, hold on a sec.
Oh, I just turned it off.
That's okay. No, it's still there. Okay.
Uh,
Maybe I'll close this again though while you're doing that.
okay, Doppler's off.
I'm just gonna try
Yeah,
to.
reboot with the Doppler on maybe. Or with the Doppler off.
Are you coming back a bit? No. Okay. I just came down a bit, so wing rate, so
Okay.
stretched.
Yeah, I'm flying off the Atalanta camera, so.
Cool. Perfect.
What if it just worked this time?
That would be great.
Thanks, yeah. I'm full of good ideas like that. Okay, ready for Doppler?
Yep.
Doppler's on.
Okay.
I see the little dot.
How can that be?
Did the dot appear when I turned the Doppler on?
Yeah. I mean, we could just navigate with the USBL. It's just going to be jumpy.
We need the Doppler for the survey. Generally speaking, if this was a normal dive, I'd be totally fine with the USBL alone. But that's our nav data.
Yeah.
The Doppler's giving good data.
Hold on, I need to talk to Rennie.
Okay. I'm backing towards you.
Okay.
You should, 25 meter delta is totally fine though. Did you just send a reset that worked?
Okay. Looks like I was missing a step that it's not clearly written in the checklist for me anyway.
Okay, what's that step?
It was changing this from cursor to USBL.
Okay.
Like I'm changing this one, but not that one.
Okay, what was it set at? Cursor?
Yeah.
Okay.
So I guess it was wherever the cursor
Yeah,
had been
I'm
before.
used to the cursor being a plus. I didn't realize that's what that little green dot was the cursor.
I didn't either. Okay, we're good
Okay.
now.
Yeah,
Sorry
sweet.
about
Okay,
that.
I'm caught up.
Okay.
Great.
So yeah, I was thinking for moves, 162 would be good.
Okay, we're set up well for that. Let's do, we've got our Doppler. Let's do a white balance. If
Yes,
you're ready,
let's
video.
do a white balance.
Sweet. Do you want to set us up for a white balance?
Sure. Do you just bring the arm?
Yeah, you want to get the white on the shoulder that you noticed to be in the light and just sort of in front, like elbow up.
Yep. Do you know how to tell that?
Do you retract the camera for that?
You just, because you want the elbow out in front where it's going to be well lit.
Yeah, okay.
And then
It's like somewhere like there?
yeah, that's
Good.
perfect. And then you center up with the pan and tilt, and then video can go for zoom.
Okay.
And you're halted.
I'm halted.
Yeah, video, go do your thing.
Thank you. Okay. I guess I don't need to do a cinematic zoom for this.
It's good practice.
Trying to get the focus. Yeah. So I want to get that. Okay. Oh, so here. Okay. Is that two? That's. Okay. I went pretty far down.
Start there. Okay, so tell her you're going to do the black bounce first.
All right, I'm gonna go for black balance. Is that okay?
Yeah, do it.
Okay.
That will never not make my gut clench.
Yeah, it's pretty terrifying. Okay, and go for white balance. Like zoom out. Yeah, turn the arrows up. Okay, and then I hold this one, right? Okay. Awesome. Thank you, Amber. That was not as complicated as I thought it was going to be.
Okay, am I good to move the arm?
Yes, you're good to go. I think we're done.
Thank you very much.
Okay, Pieta,
Yes.
what's up? What's your first request?
If we can get a Niskin at the bottom, but I don't. Is all this sediment caused by us, or is it there? Can we move a little bit away from?
Yeah, we can
'Cause,
try.
yeah,
Totally. We'll see what I'll see if we can get away with from it. You don't want the sediment?
possibly not.
Okay.
Especially if it's from us getting to the bottom. I'm not sure why. If
Okay, sounds
yeah.
good.
Just a little farther.
I'm gonna back down and then move a little bit to port to
Okay.
see if this is us or
Sounds good.
just
might just
what's
be
here.
Yeah, looks better now, huh?
Yeah.
Okay, anytime, whenever you want.
Okay.
Yeah.
Interesting line on the seafloor, isn't it?
Always, yeah.
Can you come down just a little bit?
Yep.
That's nice. Noelle?
Yes.
Are we good to go for Niskin number 1?
That would be perfect.
Video, can you go full wide, including vignettes and everything? Thank y'all.
No problem. Give you a little more iris too.
Thank you, great. And Noelle, can you keep an eye on the Niskin cam?
Yeah, that one went.
Yeah. Okay.
3008. Okay. Thank you.
Am I good to push past the vignette?
Yes.
What is the depth here saying? 3018, and there is like 3008.
I don't know.
No.
Which two depths are you comparing? Yeah.
This one, when she logged it, and the one that was there, that's 3008.
Oh, that is interesting. Do we have two different depth sensors, 10 meters apart?
Not sure.
Are you talking about the difference between depth in sea log and depth on the vehicle?
Yeah.
Yeah.
Yes, that is a thing.
Oh, okay.
I cannot remember why, but I know it's a thing. The depth that we get on the vehicle is the depth that the vehicles are. There's a small difference between, like the CTD is slightly different depth than the actual depth sensor, like a meter apart. But I don't know why sea log would have a 10 meter difference. You said a 10 meter difference?
Yeah.
It's 10 meters difference. Yeah, I could see a couple meters, but 10 seems a little wide.
10 seems
Maybe something needs recal.
It's possible that that's coming from the USBL depth.
We can check that. USBL, can you bring up Sonardyne again? What's the depth you're getting for in C-log?
3018.
Yeah, that tracks with USBL depth.
Yeah.
So
Yeah.
I'd go with the actual
Yeah.
depth sensor depth.
yeah.
rather
So
than
I
the
always, because
interpolated.
if I'm not here, I always do -10 from the
Okay, you can go back.
from there.
Well, we
I
don't
think
know
that's
that
just
that his.
this
Yeah,
Or
that's
it's just
right
this.
now.
It's not going to be a 10-meter
consistent.
difference every
Oh,
time. So
okay.
you,
Just
if
use
you
that.
have the Pero in front of you, if you have the vehicle depth sensor in front of you, that's the one to use.
Oh, no, yeah, I know. I'm just saying if I'm not right here, I need to ask the other guys to check the actual depth when they trigger the Niskins.
That's a great question.
Yeah. No, because I noticed that in the previous days that Meringue was telling me a depth and then on C-log was a few meters different. So I thought she might have made a mistake.
Okay, so
or I
you
don't
want to
know.
do 160,
But,
you
My
said?
guess is in the grand scheme of the depth
Yeah, I mean, it
sections,
doesn't.
it won't matter.
Yeah, it won't matter.
For your
Yeah.
interpretation of data.
It won't
but-
matter at all.
Science, are you ready to get moving? It's going to take a little while to get the vehicles going.
Yeah, let's
Sure.
get the photogrammetry going first.
Okay,
So
okay.
Noel, are you ready for me to go ahead and do the zoom mid and things like that, or what do we do?
Yes.
And
I'm-
we're ready for you to make the camera adjustments that you need.
Yes?
I'm ready.
Okay. Zoom mid. Okay.
That's correct. And Gabby, our instructions are to ask ROV to set upper cam zoom at -6.
Copy that. Is upper cam stereo cam, right?
Yeah, probably.
Yeah, there we go. -6.
Thank you. And we are setting to
I'm
zoom
still
in.
working on the
Okay, sorry.
getting it dialed.
And Jeff, our autofocus is on. It should be great.
Autofocus is on, yes.
Yep, great.
I can't get it to -6. I can only get it to -7, if you can believe that. -7 or -5.
Of course.
Okay.
Just fluctuate between the two.
Yeah. -7 is the closest I can get.
All right. That's going to have to be fine then.
Okay.
Um,
Are you ready to toggle timer?
I think we're ready.
Okay. Starting the transect, photogrammetry transect now. It is flashing red at me.
Are you seeing the number go up, down on that bottom part?
No.
No. I'm gonna go look and see if it's starting to take images.
Let's see.
No new images are showing up in the folder yet,
1 minute.
but there may be a write delay.
Flashing red doesn't seem promising, but
Oh.
let's see.
It is capturing. It does say it's
So-
capturing.
It does say capturing on the screen. I think we're doing it.
Okay.
We should see a number going up, Jeff, on that
On
bottom.
here, I see it says upper 0 to DL, cinema 0 to DL.
That just means they're all downloaded because they're
Okay.
coming up over 10 gig fiber. So they're like here
Super
before
fast.
they've even been taken.
Okay, let me double
Not
check.
really, but
Yeah, your point's taken. So, I'm looking. Okay, I'm getting.
Hold on, Jeff.
Okay, I'm finding the images now, Noel, in some of the other folders.
Okay, sorry, Jeff, what were you saying? You can see something?
I'm seeing the stills are in the timer still PNG folders, and I'm seeing them now. They're there.
Okay. Then
Okay.
I think that's it.
All right, let's head down closer to the seafloor and start heading up slope.
Sounds good. Yeah, you can put that moving.
What do you think? 30 meters?
Yeah, sounds good. I think we'll probably want to keep them going, though.
What speed do you want?
0.3. It looks like this is going to be pretty
Bridge, nav.
straightforward for a while.
Could you please do a move 30 meters, bearing 162 at 0.3 knots. Thank you.
And just a heads up, the bottom cam, I think you call it the cinema cam, is not in focus.
The cinema cam is not in focus. Okay. Should be in autofocus. So maybe we need to toggle that off and back on.
It might do better once I'm closer to the seafloor, though.
Okay.
So I'll come down. But I have seen it struggle with this before. There we go.
That looks better. Jeff, if that number climbs and continues to climb, if you notice it during the thing, yeah, so that's what it should look like.
Mm-hmm.
If you see those numbers just continuing to climb, that's a problem, and we need to let DE know. So just check in on that periodically.
Yeah.
Are you managing to get them all uploaded pretty quickly?
Yeah, it seems like it.
Okay,
It's just at
awesome.
1 and dropping pretty fast.
Yeah, that makes sense. As long as we have the fiber pathway, we should be fine.
Great.
Yeah, lightning speed. I love it. Yeah, Mike, I've got the Ethernet bottles off right now, so but we have the capability now to fail over to the Ethernet bottle if we lose the fiber line.
Jeff, you are
Is
also
that a
still
fish?
able to take stills on that camera if you
Okay,
want to.
so I can still hit the take photo button.
Correct. You then just, well, you might need to scale back out to mid-zoom if you zoom in or something.
I think I'm not going to mess with it.
Okay. I think that's a good call.
Could be.
Smart.
Is it? Sure.
There is a fish there.
Oh,
Is that a fish, this thing right here?
I think so. Ooh,
hold on.
switching it up.
Yeah. That.
Is that a fish?
I don't know.
It looks very fishy to me.
I think it's a stick.
You're supposed to laugh. That was kind of funny.
I'm smiling.
Okay,
I made my lip curl.
that's about what it was worth. Go for zoom.
Lip curl. My eye twitched a little bit.
My eye is twitching all the time.
That is not a fish.
What?
What is that thing?
Is that a rock?
That's a rock.
Oh,
Nice.
Disappointing.
Definitely.
Oh,
It's
to
a
a great
very
start here.
Fish-like
A
rock.
yeah,
though.
rock
Go
shape.
wide.
Yeah.
Super fishy rock.
Is this just a track in the sand in front of us? Or is it a...
Shrimp.
Oh, there's a shrimp.
Yeah, let's go check that guy out.
Okay.
I don't see a line, if that's what you're
Yeah.
wondering, Rye. But your eyes were better than mine yesterday.
Where are you looking?
Just kind of top right of the screen, there's a line. It's just like, looks like a sand
I'm gonna go take a look at that real quick.
track.
You can see it better in lower WCA, but
Okay. Only when it's in focus,
I don't
though.
see a line in it.
yeah, okay. Yeah, it's not as straight as it was yesterday.
Oh, I think I see what you're looking at, but it's this like track down.
Sand
Yeah.
track?
Yeah. Sorry about the shrimp. I can still
No worries.
see it.
Yeah.
We'll be able to look at it. It's
Well, and there'll be more of them too. So
there will be more.
It kind of looks like it was made by a stick sledding down the hill.
Snow day.
I hope it had a good time.
Yeah.
Maybe it's some abstract sea cucumber art.
Yeah.
I think the shrimp is coming. Yeah, shrimp came back.
Oh, perfect.
Do you want me to stay here or keep moving?
Oh, keep moving. It's gonna take a while to get these things moving.
Alrighty.
Go for zoom.
Bridge nav. Could we please do 50 meters at 162?
Oh, great. Okay. There's a whole bunch of these shrimps, and they're all red, and they all look really, really similar. But this guy, I think, is Cerataspis. You can actually see it's got like a sword, like rostrum, sticking out between those
Go what?
two big plates on its head.
Like 10 meters away from it.
What are the plates on its head for?
They're called antennal scales, and they have a bunch of sensory structures on them.
They're like radar dishes.
Okay, go for zoom.
Probably gets better Wi-Fi than I do in my room.
That's great. So these guys are actually big predators. You can see their claws hanging down, and they're kind of probing along in the sediments, and they eat all kinds of things. We've seen them at baited cameras, and at baited cameras you get lots of these little crustaceans, white crustaceans showing up called amphipods, and then these big shrimp will come by, and instead of eating the bait out in front of the baited camera, they grab the amphipods and swim away with them. You can almost hear the amphipods screaming. It's just awful.
All right, so the latest saga is I'm not getting a trail at all from your DVO.
Okay. What's the trail length set at?
5.
Oh, I think it needs to be longer than 5.
Let's try, what do you think, 20? 20?
Yeah, 100. I wonder why it's set up. What changed in the setup here?
I don't know. Sure, I could make this bigger too. I'm not sure what it usually is.
That's just the marker size, I think, right?
Yeah.
There we go. Got a few.
All right, that looks better.
Sweet. Sorry, video. Every time I look away from the screen, I change your lighting for you.
You've been changing the lighting?
Well, no, just coming up off the bottom.
Oh, yeah. No, you're good.
Okay.
It's my job.
Yeah, you're doing great. Oh, C-pen or something, some other biology. Go presume.
Everything is either a C-pen or not a C-pen. So.
Yeah. I've pretty much nailed taxonomy. Yeah, it's not a C-pen. Could be like anything else, though.
It's a 50-50 chance, so.
Can we get the lasers on, actually?
Yeah, I got them.
Go wide. My taxonomy is pretty much foolproof.
C-pen, not C-pen.
It's a two-step dichotomous key.
Thanks.
That was great.
Biodiversity explained.
I used to go with squishy versus crunchy. That's pretty good. Which is really good, but that actually contains real information. And C-pen versus not C-pen is not actually
Less
increase
useful.
much of the information that you actually have.
That's about as good as we collected what we believe to be a sea cucumber on a dive the other day that was white and squishy looking. And in the lab we had, all the science team down there, everybody's looking at it, and the closest we could get to an identification was animal.
Perfect.
In our defense, we did refine it.
We did refine it later, but
It just
that
took
was
a long time.
after the dive. That was all we were confident in.
Okay, so what did you get it down to?
We do think it's a holothurian.
Okay.
We also think it is likely a new species, but it's hard to say. We are trying to find a deep sea sea cucumber expert. Believe it or not, not that many of them.
Nope. And we know what family it's in. It's in
We
the
do
family.
know the family.
Yeah, Solidae. So.
Okay. Derek, what have you done?
This wasn't set up right. It's all right. We'll just go with this.
Yeah.
It's fine. You just won't see the graticules on the right.
The what now?
It's by lat long instead of meters.
Okay.
It's...
Huh. Okay. Sorry, I know I'm in the cabbage a
That's
little
okay.
bit here.
All right, I'm going to try to reset this then.
And also, Derek is doing a great job. I was just hassling him because DVL nav is, or NavG is acting up.
Noel, do you know how to change
love
that
it.
screen there so that it's the
I don't know.
the nav? I presume it's something in here.
Map projection's very finicky, so I can...
Oh, that is
close
my
it and
monitor
start
left.
it
I
from
thought
the
this
start.
was me here.
Oh, you're
Okay.
in lat long now instead of Alvin XY?
So monitor left.
Yeah.
So
and
you're usually
then I
in like
can find the
UTMs or Alvin
nav.
XY or something?
ROV
Yep,
nav.
that's what I tried, was Alvin XY, and then it got really wonky.
What about the one with the map?
Okay.
Oh, high-pack survey. Beautiful. Thank you.
Well, I could reset them all.
Yeah, if you could just like each one of these, just one by one.
Well. Oh, you're kidding. Okay. You're messing with me.
I'm just trolling you.
It's because there aren't enough like rocks and samples going on right now to keep me out of trouble.
ROV pilot's board. Just coming in minutes.
All right, I'm going to move on.
I think it's great. Like I can fly by it, so as long as you can operate by it, I think it's fine.
Okay.
Have we ever-
Bridge, nav.
Oh, yeah.
Is that another cucumber on the right?
Can we please
Yeah,
do
let's go
50
take a look at
meters,
that
bearing
guy.
162?
go for zoom.
All right, so this is Amperima or Peniagony. This is that one with the vellum on the head that we see swimming around occasionally. Great. Let's keep heading uphill.
Roger.
You got a fish ahead of you, I think.
Cool.
I can see it on the lower.
Are you sure
WC.
it's not a sea pen?
No.
Go wide.
It is not a sea pen.
If it's quite swimmy, it's probably not a sea pen.
It's
That's nice.
It's in the water column a bit, I think.
Okay.
Yeah.
This new taxonomy makes my job so much easier.
Might put you out of a job, actually. But have we ever had a sea cucumber enthusiast science?
I have never met one before.
I would enjoy that. Only if Derek was here, though.
Would they get along?
So we could just like...
Would we get along? I don't understand you. What
Wanna,
makes you tick?
I just want to hear his like long suffering sighs in the corner.
Do I do that? I hope not.
No, you just kind of look neutral.
I think we all do that.
Yeah. There's definitely a lot of front row sighing. I feel like I'm definitely guilty of that.
I feel like it's like snoring, like you won't know that you're doing it. When
Yeah. I usually know after I've done it. And then I'm like, ah, crap, I did that out loud. Yeah, just like with snoring, you don't know until you wake yourself up with your snores. Like, ah, it's already been done.
Yeah,
It's too late.
exactly. So I think when I restarted the whole thing, it made me have to go back.
Oh, to
like
the
I
beginning.
have to go back and reset everything.
Oh, gotcha.
Like manually type it in.
Oh,
So
that's a
I can
pain.
do that. Because I think the rest of the dive, they'll be confused why it looks like this.
Okay.
And it doesn't look like we're tackling anything too crazy right now.
No, you're free to do whatever you need to do.
All right.
Yeah, sorry, I should have caught that too. Just the cursor looks different than it used to. It looks like a pixel. It's not much of a cursor.
Oh, there's a fish on Atalanta, I think. The Atalanta can.
Oh yeah, just a little one.
Hey, Control Room, any chance you can pan left on Atalanta? There was a large squid.
Oh.
Did you catch that?
No, I did not. Oh, well, yeah.
Pan left on
Okay.
Atalanta. You're good to go.
Can try and go wide
How
with
much?
Atalanta too. It's all
Maybe
wide.
see if you... Brian, can you let us know when we've gone as far as you think you need to?
Yeah, will do. I'm not sure we were even fast enough to get it. Took us a second to realize what we were looking at.
Yeah. Okay. Yeah, Atalanta doesn't pan all that fast. You're kind of looking at Atalanta's speed.
Yeah, totally understood. I think we probably
Just
missed it,
keeping an eye
but
on this
it looked
other
like a
squid.
big fin squid.
Okay.
Ah,
I think we missed it.
Okay. I'm gonna head back.
Let's look at this coral right here.
Okay.
Are you in auto XY?
I am not.
Okay.
That looks
Go
like
for zoom.
Bathypathes.
Old friend.
Yep.
man, I'm sad I missed that squid. Did anyone see it? Just Brian.
I did not see it.
Yep, that is Bathypathes.
Okay, go wide.
Harold saw it too. I promise I wasn't hallucinating.
And you called it a big fin squid?
I know one of the big fin squids is the one with the super long arms and tentacles.
Oh,
yeah,
really?
It looked like a magnapinid squid to me.
Ah,
Really?
I'm trying to rewind in the Atalanta footage.
That was just a minute or two ago?
Just a minute ago, yeah.
It might be worth actually turning around for that. Those are super rare.
Go for zoom. Okay.
I don't know if that's possible.
Good luck finding it, I guess,
Yeah,
but
Yeah,
It's in,
okay.
I mean, it's like in the water. I
Yeah,
don't know how we'd
okay.
go about finding
would
it.
it show up on the Norbit? Probably not.
No.
Do
because
they
it's-
have air bladders?
No, they don't.
Then no.
Yeah.
If it's attracted to the light, it might come back to us.
Probably not. Lots
Okay,
of squids
go wide.
are, but I don't know if those guys are.
Here, I'm just getting my heading back to what it was.
mind if I bump up the iris
Okay.
on Atalanta a bit? Might be a bit overexposed, but just would
Yeah, whatever.
really love to try and maximize our chances of seeing that squid again. Oh,
Ooh, wow.
Go for zoom. And then after this, I have to get back out in front. That's good right there. Okay, I gotta go.
Oh, bummer.
What do you need? More zooms?
More zooms, and
Okay.
particularly in on the polyps or close to the base.
Okay. How's that?
That's great.
we have more zoom too.
Thank you.
Okay, go wide. It's time to scramble.
The big sponge on the bottom right corner. Stock sponge.
Oh, yeah. When I drive off of the upper cab, I'm like, the vehicle keeps getting stuck.
Oh.
Because it pauses every time it takes us a
Oh,
picture.
yeah.
Okay.
You're like, man, I'm bumping so many rocks.
Yeah. Bonk, bonk.
It's not distracting at all.
Nope. I'll get used to it.
Do you want to keep letting this layback play out or keep moving?
I guess maybe the way to do it is to just pause,
I think that might
let
have
the
been
ship
the old
stop at
base
the end
of
of each
coral
move
or
and then
sponge.
do the next one.
Yep.
What's that, Jeff?
Oh, Pierre and I were just talking about something that we were seeing
okay.
there.
Okay. I've got a little bit of room to work with now, so we can stop for zooms and stuff.
Okay.
So we've been paused for a while.
Okay.
Looks like they're modifying the heading slightly, but
Okay.
I'll just wait till they're done tinkering with the heading, I guess.
Yeah, then keep going.
Yeah, it seems like they're just adjusting for current, making fine-tuning of the ship's
Okay.
heading. Yeah.
What did you just circle there? Can you do it again?
That little white guy.
Okay. Go for zoom.
I'm feeling ravioli.
Is that a snail?
Looks like a shell of some kind. But like broken maybe. Yeah.
Huh. Okay. Yeah, looks like a piece of shell.
Okay, go wide.
Can I reset you?
Yeah. Does it seem like the slope is going to be pretty consistent for a while?
Yeah, it's consistent throughout the whole dive according to the bathy.
Okay.
I mean, sure there's some steeper sections, but... Bridge, nav. Can we please do 30 meters bearing 162?
Oh yeah, I see it. Go for zoom. What is that?
I think that's a cup coral.
I think so too.
Yeah.
Yeah. It's pretty big, actually.
Do you have what you need?
Yes, let's keep going.
Okay, go wide.
Is that a big stocked sponge?
Oh,
off to our
yeah.
right?
I almost completely missed that. Thanks, Noel. It's huge.
Wow.
Oh, wow. Look at that.
Very tall.
Yeah.
Oh, that's beautiful.
Wow. This looks like Calyx or something like that. One of those sponges. Whoa.
They're so iridescent. It's really neat.
Yeah, they're beautiful. Can we see anything
Go for zoom.
growing on the base? this
You want
time?
a zoom on the base?
Oh, I'm just curious.
Yeah, I'd zoom on the top part
Wow.
first, and then
Okay.
yeah.
That's stunning.
Stop there.
That is very large.
Fantastic.
Okay, I'm going to go look at the base now.
Yep. It's interesting. It looks like there's an old dead base right next to the existing base.
Yeah. Okay, do you guys
Great.
have what you need?
We do.
Okay,
Do we
go
know
on.
what type of sponge?
It's called Saccocalyx. It's a glass sponge.
Glass sponge. Gotcha.
Nope.
Oh, that was a fish.
That was moving real fast. Oh, there it is.
Oh, there we go.
Oh,
Fish.
yeah.
That looked like it might have been Acanthonus. Where did it go?
Away, apparently.
Yes, it went away.
That was very fast. It did not want to hang out.
No.
You thought that might have been Acanthonus, Jeff? Is that what you said?
Yes. We saw a bunch of them on the last dive. Let's zoom on that coral.
Sure thing. Go for zoom. Can you go wide?
Great.
Not cinematically, just go wide so that I can
Yep.
push back
Probably,
in.
probably a bamboo coral.
There we go. Now zoom.
Lovely.
Oh, that's nice.
Yeah, I think I see junctions right down there.
There is a fish right to your
Okay, go wide.
left.
Yep, let's look at the fish.
Right there. Right above your lasers.
Oh, yep. Okay.
Oh,
visit.
that's the same guy.
Oh, yeah. There's another fish on the bottom.
Oh.
Yeah.
Basozetus.
Oh, it's happening to you too, Max. Go for zoom. Starting to know all these names. Maybe you always knew these names.
Nope. That's just the one Jeff says a lot.
Yeah.
Yep, that's Acanthonus. I just need to get a zoom on the head to be certain. Hopefully it'll
Sorry about
bump
that.
into
there.
the seafloor
Gabby,
and
I
turn
think I gotta
here.
jump on the zoom.
No, it's okay. I keep bonking rocks, so it's going to make it hard for you to do anything good. Okay. You can push in.
How
Yep,
big can
there it
these
is. I
get,
can see
Jeff?
the spine on the tip of the snout there.
Okay.
Acanthonus armatus.
Okay, go wide. I'm making myself seasick here.
Acanthonus armatus, gotcha.
How big do they get, Jeff? Do you know?
Not huge. I don't think any bigger than about 50 centimeters or so.
Okay.
Bridge nav. Could we please do the same move? 30 meters, bearing 162.
Jeff, is there a term for the...
Oh, what was that right back there? Sorry.
Yep.
Just flew by.
Putting on the brakes. Was it a fish?
No, it was some sort of invert.
Okay. But it was a movie, sort of swimmy sort of thing.
It was not a sea pen.
Okay. Thanks for clarifying.
And of course now I don't see it.
Yeah, I don't either. Okay.
I'm sorry.
Oh, you don't have to be sorry. I'm sorry I didn't catch it.
Too last minute to ask for a stop.
Oh, I see it. It's on the bottom.
Yeah.
I think it blew down the ridge.
Where is it?
Directly on the bottom of your
Directly
screen.
down, yeah.
Look down.
Okay.
Yeah.
Oh, yeah.
Oh. What the?
Oh.
Maybe.
This guy right
zoom?
here.
Yeah.
Yeah, that's a hermit crab.
Oh, is it?
Yep, I think so. This is the one that's going to have a multiple anemone on its back.
Really?
Oh,
Yep,
yeah, it
this
does.
is called Sympagurus. Yep, and all the polyps on its anemone are all closed up right now because of the turbulence that from our thrusters.
Okay. Oh, that's funny.
Oh, that's
Okay,
yeah,
go
that's
wide.
pretty cool.
That is really cool.
So many of the hermit crabs in the deep sea actually use anemones on their backs instead of just a snail shell. When you get really deep like this, we are at a point where a calcium carbonate that makes up most snail shells typically dissolves. And so these hermit crabs have come up with a solution to have something else for their shell. Anemones.
Jeff, I was going to ask, though, there's like carcinization for crustaceans evolving into crabs, but is there a term for all fish evolving into eels?
Is that a thing?
Jeff was talking about it the other night. Deep
Max,
sea
he can't
fish.
hear you.
Ah,
Sorry,
That's
I'm
so
off in the
unfortunate.
cabbage.
Jeff, can you hear me now?
Get out of
Yeah.
the cabbage.
Yeah, get out of the cabbage.
Go ahead.
I was wondering if there's a term for the convergent evolution of deep sea fish into eels.
Yeah, so lots of deep sea fish have these really long eel-like body shapes, even if they're not eels. Like that fish we just saw, Acanthonus, is a cusk eel, which is a common name, but it is in no way related to eels, but it has the same body shape. No, I don't know that there's a term. There's a term that describes the swimming of that body shape. It's called anguilliform swimming, which is a mouthful, but anguilliform just means eel-like.
What makes an eel versus a fish?
So eels are a very ancestral group of fishes, and they have a particular larval stage called the leptocephalus, which means leaf-like, and they're these flat, elongate larvae that you can see straight through, and all the eels have that. And they also lack pelvic fins. They have pectoral fins, but I think maybe there might be one or two exceptions, but they don't have pelvic fins, and that sort of links all the eels together.
And what makes an eel versus a sea pen?
Oh, this crowd.
Just trying to keep you on your toes.
Uh-huh.
You got to stay at the cutting edge of taxonomy.
Yes.
It's your work. It's your life's work.
Eel versus CPET.
I think I'll stick with my mouthfeel taxonomy, actually.
Well, Jeff, it seems like an opportunity for you to coin a term for all the fish starting to become eel-like.
Eelification, something like that?
Yeah.
I don't know.
Or anguillicization, I don't know. Either would be good.
Yeah, everyone gets a huge kick out of the fact that crustaceans all become crab-like in body plan. That was everywhere on the interwebs for a while. Maybe people will be similarly interested in everything becoming eel-like.
That's what I'm thinking.
Yeah.
Bridge nav. 30 meters, 162.
Thanks, Derek. I think this works out pretty well.
Okay.
It seems to. I don't know. What do you think?
Yeah, I'm liking it.
Okay.
Seems controlled. Like if we need to stop, we can.
Right. And you're just waiting a little bit in between each move?
Yeah.
Yeah, and I don't feel like I'm having to sit around too much and kill time.
Is that something? This one right here? This black.
Something right in there.
Oh, yeah. It's not a sea pen. Go for zoom. I think it might just be a rock shadow, actually. Go for zoom.
It would be really funny if it was, actually.
A sea pen?
Yeah.
Yeah. That would be good.
Is this a rock?
Yep.
Go wide.
Yeah, I'm playing tricks.
What is that?
Oh, yep.
Another hermit crab?
That looks like another hermit crab.
Oh, nice.
That guy is
It's running.
booking.
Just moving.
He's running late.
Yeah, again. Go for zoom.
Oh yeah, the
Yep.
anemone on
That's-
the
Yep.
on the
Hermit
back.
crab. Hermit crab with anemone. Can we get any closer zoom?
Yeah, we probably can.
No closer zoom, but we could
Go wide. Maybe closer vehicle, though.
Okay.
Jeff, how do they acquire their back anemone?
I do not know. Isn't that a great question though?
That's wild.
I mean, they've
Is
got
it
to.
just
How do they start off
Do they
and
farm
not?
it?
Yeah. Or is there
Zoom in?
something about the back of the crab that causes the anemone larva, planula larva, to find them? How does that work?
Is it always an anemone?
Okay,
Do we
that's
see
good.
any other?
In these particular groups, yes, and it's almost always the same species of anemone.
Really?
Yeah.
Interesting.
There's some variability, but a couple of taxa, the crab always has the same anemone species.
That is wild.
I am not sure which one this is. This might be... Yeah, I'm not sure which one this is.
I think that is heavier or lighter than carrying a shell around all day.
Okay, I should probably get
Later.
going.
Okay,
Go wide?
thanks. That was great. Very fast hermit crab.
Oh, sponge.
Oh,
Yeah, another Saccocalyx.
Go for a zoom. You can go in a little faster than that for these.
Bridge
Great.
nav,
Perfect.
30 meters, 162.
That is great.
Okay, go wide.
Thanks.
That looks steep.
This is really steep. Derek, do you know what the slope is here? Does the map tell you that?
I mean, on the map it's about 28%, but it doesn't resolve this kind of micro topography.
Can we check out that coral right there?
Oh yeah, I don't even see it. Wow. Good eyes. Go for zoom. That's good.
If Brian is online or watching, maybe he can tell us what this is. This looks similar to one we collected yesterday, but
Oh, yeah.
I did not find out what it was.
Okay, go wide.
Thanks.
Also, if we're going to sample around here, that decision needs to be made like pretty quickly.
Fast.
Yeah.
Yeah.
Because we won't be able to like stop the ship unless we plan
we'll
to go
have
back.
to come up,
Yeah.
right? Yeah.
It'll be just sort of like a
flying
sample, though we're about to get off watch. That will also apply for our next watch too.
Sea cucumber left.
Oh, that's a big one.
If you look even more to your left, is that a coral?
What do you see? Ooh, eel. Yep, that trumps sea cucumber.
Are you sure?
Go for zoom.
Dust storm coming.
I know, I see. It's terrible. Not my best work.
Okay.
Dune.
This is a cutthroat eel.
Go push in further.
Oh.
Okay, go wide.
And we're dusted.
We'll just keep an eye on it and see if we... It doesn't like the dust either.
Nope.
Okay, push in. You need the head, correct?
Yes.
Okay, stop there. Oh,
oh, nice.
Nice swirl there.
Maybe that was a little chrysogorgid coral
Push in.
there, and oh, this is great. You can see the whole dorsal fin insertion.
Ugh. Every time I bump a rock, it gets
Yeah,
scared. Go
right.
wide.
No, that was pretty good.
Okay, cool.
Thank you.
Yep.
Oh yeah, he's gone.
Nice fish chasing.
Yeah, that was great, Gabby. Very cinematic follow with the
Yeah,
ROV.
that was cool following the dorsal fin like that. I'm gonna come up pretty fast now.
Okay.
Trying to make a beeline out of the cabbage.
No rhubarb
Oh,
today.
cool little fish right there.
Rhubarb, cabbage, yeah. Trying to get back to that little fish there, Jeff.
It's okay. I'm sure it is gone.
Okay.
It was really close to us by the time it came on camera.
How's the saying go? Out of the cabbage and into the rhubarb?
Yeah. Out of the cabbage and into the frying pan?
There you go.
Little fish
Oh,
right
another
there.
fish.
Oh, yeah. Okay. Go for zoom.
So tiny.
That is Basacetus. Okay, we can keep on going.
Okay, go wide. Does Basacetus ever get bigger than that?
Well,
Or is it just a teeny little
there,
fish?
No, there's a bunch of species, and we keep seeing these really little ones, and I need to actually x-ray the specimen that we have to really ID it with confidence. Either there's a lot of juveniles around, or it's just a particularly small species. I don't know.
Gotcha.
But at baited cameras, when we get down to 3,000 and 4,000 meters, the Basocetus that we see there, which we think is Basocetus nasus, is often a meter in length. It's big.
Oh, wow. Okay.
Really big.
So pilots, new heading is 168, by the way.
Copy that.
Just slightly different.
Jeff, I finally figured out how to spell Basocetus, and I have spelled it incorrectly probably many different ways every time I entered it until this moment. There's a Z in there.
Yep.
Did not know.
I may not be pronouncing these scientific names entirely correctly either.
I'm usually okay at spelling, but that one really got me.
Bridge, nav.
What's up?
Pilot, we're 15 meters from completing a 25-meter move.
Okay. Roger that.
Our heading's 168 for the ship moves.
168.
Oh. The bearing for the move.
Oh, cool. Let's check out this Venus flytrap anemone. Looks like it's on a dead coral or sponge stock.
There's something on the stock.
Can we get a
Is
zoom?
it?
Yeah, you can
and
push in for a zoom.
Maybe not. No, maybe not.
we want to check out actually the stock below the anemone as well.
Okay.
Something red
Yeah,
and white.
that thing right
That-
there.
Okay.
I'm not sure what it is.
I don't know. It might be a zoanthid or a little anemone of some kind. Wonder if it's a juvenile Venus flytrap anemone. Hard to tell, huh?
Yeah, not sure.
Okay. We can keep going.
All
Thanks
right.
for that.
Yep. You can come wide, video. Thanks.
All right, to our viewers, it is time for a shift change. I would like to say thank you for exploring with the 8 to 12 watch. We will see you next time. All right, send in your questions if you see anything we might have missed, or if you're curious about the team or what we're doing on board the Nautilus. Please put your questions in the chat. All right, 8 to 12 signing out.
Oh. Keeps me sharp. Have a look at this star. Is that what you said?
Yes.
Okay.
Looks like Polyasterius.
Push in there.
It's pretty cute.
Yeah.
That's perfect, thanks.
Okay. Come back wide.
Bridge nav. Can we do a 30-meter move, 170? There's another sack of calyx, or is that the same one?
A different one.
Different one.
Yeah, we've been moving.
Oh, I think it has little ctenophores on it, if you want to snap in. See those little benthic ctenophores?
Push in a bit there. Oh, yeah.
Remember on the first dive we had a bunch of those purple ones on the Chrysogorgia?
Yeah.
It's like that, but this is what we typically see. These are called Gelfiella.
Gelfiella.
Gelfiella?
Yeah, but it's got a silent T.
Okay, that's great. For my spelling skills.
I never would have known. So.
Yep. Yeah, it's like TJ, you know.
Not that I'll ever try to spell it, but couldn't tell you where the silent T was.
Well, there's a little single polyp sclerotinian, like the one we sucked up the other day.
Oh, yeah.
I hope we see another big fin squid.
What? Someone saw a big fin squid?
Yeah.
When
was that previous watch?
In the previous watch. It was in the Atalanta view.
Oh, fish.
Oh, yeah.
Nice.
What kind of fish are you? I think it's a rat tail.
Push in a bit there.
See the dorsal fin? That's how you tell it's a rat tail versus a cusk eel.
Every time I get one right, like in my head before you say it,
You're
I'm
like,
yes.
Oh, that one's really cute. Kind of looks like Nazumia, but this is deep, so maybe something else. Maybe a smaller Coryphaenoides.
All right.
We have a question about eDNA. So the viewer is asking if the Niskin bottles capture the eDNA, how do you stop human DNA from contaminating the samples when you handle them?
Is it a squat lobster?
Yes, it is.
So we go through like a very
unidopsis.
rigorous process of decontaminating our stuff. So whoever is touching the Niskin wears gloves, and whoever is touching the Niskin does not touch the bottle. So either me or Piera is touching the bottle, and we are also wearing gloves. But in between touching stuff, we spray our gloves with RNAs to remove any human DNA before we can touch our stuff to minimize contamination. Thank you.
Yeah, now that we're kind of settling in here and within a watch change, just wanted to go a quick recap here for those viewers who might have joined us recently. So welcome. Thank you so much for joining us. This is the 5th dive of the Leboo's voyage expedition aboard EV Nautilus, exploring never before surveyed in the Palau National Marine Sanctuary, supported by NOAA's Office of Ocean Exploration and Research. Today's dive, we're diving on a seamount that up until this morning had only been partially mapped. In fact, Nautilus, when it was coming, and the previous cruise coming from American Samoa over here, did a little bit of mapping around that, mapped about half of it, and then this morning our mapping team mapped the rest of it. And let's take a look at that cucumber.
So the sea cucumber is a new one. We haven't seen this one yet. This is a Psychropodes. So it has this cool tail. We lovingly call it the gummy squirrel.
The gummy squirrel. I remember that one. Yeah. We can push in there.
This is really cool.
So is that is closest to us the front or the back?
That's the back.
Okay.
But sometimes their little tail is in different positions on the body. Sometimes it can be more in the middle. Well, there's a little snail.
That was a good question. I had the same one, Sarah.
It depends on what you decide the front of a sea cucumber is, too, you know?
Yeah.
It's a cool snail. You see it? The little
Oh, yeah.
swirl. It's on the move.
Yeah, it's really fast.
It's a really pretty shell. It's like iridescent.
Nice.
This is a fantastic zoom. This went right in there. What is the snail called? I don't know. I'd have to look through some of the mollusk books to see if we can get into a family.
All right, you can come wide there.
What's the point of the tail on the sea cucumber? Or tail like
it
feature?
Works as a sail. Kind of helps it glide through the water.
Oh, okay.
So these guys will feed on the bottom, and then you know how they do their crunches and they swim away.
Yep.
That just helps them glide further distances.
So less crunches, more distance.
Mm-hmm.
Nice.
And crunches are hard, you know. It makes you tired. We see that genus a lot on the abyssal plains. There are
some that are like lemon yellow too.
Oh, wow.
Yeah. That's like the original gummy squirrel. And their tail can be very, very long as well.
There's another one. Not of the one with the sail.
Yeah, this is a different genus. Looks like Oniraphanta, a type of diadematid
Quick snap zoom
sea
there.
cucumber.
Spiky.
Yeah. But if you touched it, it would be very soft and squishy.
All right, come on.
Do you know that from experience?
Kind of like a koosh ball, you know, but in the most awkward of ways.
Yeah, so just circling back a little bit on the dive plan here. So as I mentioned, the seamount had only partially been mapped, and then our team mapped it overnight. And so the plan is to go up this seamount, one of the steeper northern slopes of the seamount. So we started around 3,000 meters, and the plan is to go all the way to close to the summit. And end at around
push in there
2,200 meters.
Tough angle
for those folks that are.
So this is Romilogorgia militaris. We saw this on the first dive. Quite common down here at these depths. It's a type of chrysogorgid coral.
All right, come on. There was a big bamboo coral to the left too.
Oh yeah, we love big bamboo corals. That is a nice one.
Wow, that's pretty tall.
Mm-hmm. These types of bamboos can get really, really long. I don't think we've done introductions yet, if we want to do that.
Well, yes. Let's do some quick introductions, and so I will start. My name is yyyy-mm-dd, and I am the Science Communication Fellow for this watch.
Ali, good afternoon, aloha to all. My name is Daniel Wagner, sitting in at Watch Lead and also sailing as Expedition Leader. Thanks so much for joining us.
Hello, good afternoon. My name is Maikandi Andres, and I am the scientist for this watch.
Hello, my name is May Lubetkin, and I'm the science manager on the ship and the data logger on this watch.
I'm Megan Putz. I'm the navigator on this watch and the knower of many of the things.
My name is Jake Bonney. I'm in the Herc seat.
I am Sarah Surgeon. I am piloting Atalanta.
I'm Erin Rainey, and I'm in the video seat.
A question from the audience. They want to know if there's a geologist on this watch.
So, yeah, great question. So we don't have currently in the control van a geologist, but we well, actually, I should take that back. You are a geology expert.
Well, I mean, I don't know if I'm an expert, but I did study geology and volcanology. I'm sailing as more in a science coordination and managing of the lab space rather than a scientist who will continue to study what we collect. But I have a background in geology. But I believe we have some geologists ashore, right? From some scientists ashore?
Yeah, we do have a few, and then obviously also on the ship we have Dr. Bob Ballard, also known for many things, including for a lot of geologic important discoveries. And yeah, when we staff This and every cruise, we try to have a mix of different disciplines and backgrounds and knowledge bases on board. Now this particular cruise was funded as a direct contribution of a commitment made by the U.S. government at the Our Ocean Conference to map the Palau National Marine Sanctuary and support work within the sanctuary. Work that started kind of 2 years ago. Also, we're very fortunate to have been invited to be part of the process of developing the science and monitoring strategy of the Palau National Marine Sanctuary. A lot of a lengthy document that was published about a year ago that sets out a whole suite of priorities about what type of science and monitoring activities should be occurring within the Palau National Marine Sanctuary, and heavily focused on biodiversity and understanding what kind of species, habitats, and ecosystems are found within this very large marine protected area. So it currently covers 80% of the exclusive economic zone of Palau. But yeah, it's an interesting area here geologically that, this is not my area of expertise, but the main island group of Palau, I believe, has been dated to about 40 million years. But there's over 200 or so seamounts around. And these are likely from very different traces, so formed of different places with very different geological ages. And so one of the geological objectives of this expedition is going to be, in particular, when we're diving off these offshore seamounts, which, by the way, have never been surveyed before. Every single one of the seamounts that we're diving on this expedition has never yet been surveyed, sampled, visually explored. And so one of the objectives of our dives is going to be trying to obtain a volcanic rock that can help us date these features. And that really ties in really nicely into the biodiversity assessments. Of those 200 or so seamounts that are all around the Palauan Islands, these are likely of all kinds of different geological ages. And because seamounts are kind of like islands that are isolated, they are surrounded by vast amounts of ocean. The biodiversity, among many other things, one of the important factors of what kind of communities they have is how long they have been in place, because seamounts that have been in place for a longer time have had longer time to be settled by larvae and then inhabited. And so, yeah, that is one of the objectives of this expedition. In addition to just basic baseline information, understanding what is within this large marine protected area, there is a marine spatial planning process currently undergoing within Palau. So I mentioned 80% of the Palau National Marine Sanctuary, 80% of the exclusive economic zone is currently protected by the Palau National Marine Sanctuary. But the government of Palau, and in collaboration with scientists from many places, they don't just want 80% of protection, but they want 100% of it easy and sustainable management. And so understanding what's in and around this protected area is important, so you can protect some of the more vulnerable habitats using more restrictive measures. But in addition to helping inform policy decisions, it's also about just
push in there,
raising general awareness about what's here. Out of sight and out of mind is usually what we say, and that's great.
There's a tiny polycube up at the top. I think this one is a Bathygorgia, because all the polyps are on one side. This is another unbranched bamboo coral.
And Megan, if I remember correctly, that is also a genus that is found in deeper waters, right? I thought it was 2,500 meters and deeper, something like that.
Yeah, it's definitely a more deep water exclusive.
And so yeah, today's dive is matching our previous deepest dive of the expedition. And so yeah, we are currently at 2,850 meters.
All right, come by there.
And I was saying here in terms of the management importance of these surveys, it's one important part of in marine resource management is just raising awareness of what's there. Out of sight, out of mind is a general problem that a lot of marine resource management faces in marine conservation is when folks are unaware what's there, it loses a little bit of the importance. And so yeah, these expeditions about bringing these remote and deep places to the people via telepresence technology are really important. Yeah, maybe if we can do the smaller one, if we can take a quick look at that.
Push in there.
It's a slightly branched bamboo. See at the bottom, it's got a little bit of a branch. This one has really long intertentacular spines. There's like little spikes between the tentacles.
If I remember D1 clade or something like that, Megan, do you remember?
Yeah, well, this is typical of like Lepidopsis, to have the long spines on one side of the polyp and like slightly shorter ones.
Intertentacular.
Intertentacular.
That is a great word.
Just have to find how to use it in your everyday life.
All right, come on, yep.
Bridge nav.
So a follow-up question on the geology. Do we know what kind of rocks are we looking at?
Yeah, these these are basalt rocks. So as we're moving up slope, just due to gravity, a lot of these rocks will crumble from where they were originally emplaced after weathering over time. And so what we're seeing is a bunch of sort of broken up boulders or basaltic talus from an old eruption. And maybe as we move up slope, we'll be able to see some more in-place rocks. Looks like even up here in the top right of the screen, we're starting to see that. And then everything is dusted with sediment from the long period of time since the previous eruption. So that's kind of the main geology of this area. In the much shallower parts of this region, we've seen carbonate rocks, which is also black, but that blackness is manganese covering the carbonate, and the carbonate is old submerged reefs that form essentially like a rocky substrate and something that the organisms and biology can live on.
Yeah, one more kind of interesting fact to add here. So I mentioned that this seamount wasn't mapped in its totality until this morning. Overnight, we also mapped a feature, actually this morning, right before, a feature that's just due west of here, and that's a perfectly round seamount that also has a caldera up on top. So yeah, you can really see your classical volcano shape on that, and it was just unfortunately a little bit out of our reach to make it to this morning for on-time launching, but
oh, that's awesome.
So this is a halosaur called Aldrovandia.